I'd like to introduce Dr. Kevan Herold. He's the CNH Long Professor of Immunobiology and Medicine and Endocrinology at Yale University. And his laboratory studies autoimmune diseases, whether they occur naturally or as a consequence of immunomodulator therapy. Sort of a perennial focus of the lab is type 1 diabetes, which, as most people know in this call, results from autoimmune destruction of the pancreas. And they're identifying immune cell responsible for attacking pancreatic islets, as well as studying how beta cells respond to these attacks. And, you know, as most people also know, you have been instrumental in, I guess, ferrying Teplizumab from its very beginning to the great news the other day that it has been approved by the FDA for treatment in those who are 8 years old and above who have stage 2 diabetes. So I wondered if you might just kind of give us a little backstory on how Teplizumab first came to your attention. And I believe that it was you and Jeff Bluestone early in your career, right?
Yeah. So, when I was a postdoc in Frank Fitch's lab, I had gotten interested in identifying ways of modulating immune responses in vivo in people. And at that time, the work that was going on involved the use of anti-CD4 and anti-CD8 antibodies. And one of the interesting observations was that the observations from Herman Waldman, which was that if you gave antigen under the umbrella of an anti-CD4 antibody, that you induce tolerance to that antigen. So that was kind of interesting from an immunology point of view, but things kind of changed a bit when actually Jeff Bluestone came to the University of Chicago, and he had been interested in anti-CD3 antibodies. And so we ended up collaborating. There's a very to me, it's a very interesting story about a typical Chicago evening. His lab had a retreat at Grant Park. For those of you who've been in Chicago, you know where Grant Park is. And it was, a winter night, and it was so freaking cold, you couldn't even go outside and skate. They had an ice skating rink. So Jeff and I were inside, and we ended up talking about this project. And so that's how things really got started. So I have to thank the lousy weather in Chicago for getting this whole thing to go. So eventually he had the drug made he collaborated with folks at Johnson & Johnson that had humanized the molecule and rendered it Fc receptor non-binding by the 2 alanine substitutions in the IgG1 molecule. The heavy Fc portion of the IgG1 molecule. Around that time, Lucien Chateauneau had published a paper. We had done some work in low-dose strep diabetes, but Lucien had published a paper in NOD mice. We were just getting NOD mice at that time, and she showed that you could treat the mice at the time that they presented with hyperglycemia and, in fact, reverse the disease. So it brought up this idea, and you didn't have to continuously treat. It brought up this idea of tolerance, which was what we were very, very interested in. And then once once this drug had been made, I basically wrote an IND to do the trial. You'll know this is a long time ago because basically an investigator writes an IND and does a trial. So, it just doesn't happen anymore. So that's what happened. And the first trial looked good. And then things kind of developed from there.
Yeah. And it had a little bit of a bumpy road, correct? I mean, it was, kind of once these discoveries were made, it was a little bit, went into the back burner for a while.
Well, I'm not sure I'd say it went to the back burner. I would say it was dead. What happened was the first trial that I did was successful, and then the ITN did a trial that seemed to be doing well. And in the meantime, Macrogenics picked it up, and was going to do a phase 3 trial in patients with new onset diabetes. But the primary endpoint that they selected was not a good one, but probably one that frankly would have resulted in approval by the FDA, but not a clinically validated endpoint. So when that trial didn't meet that endpoint, Macrogenics bailed, and so did Lilly, who was providing funds for the trial. So it's interesting. Now it became an orphan drug for an orphan indication. So pretty much, I think we as myself and Lucien Chateauneau and Jeff were searching for individuals who might pick this up. So we ended up talking to a lot of people. In the meantime, I had put in the proposal for the prevention trial in TrialNet. And after a few votes, that finally moved forward. So at the time that we were getting the prevention trial, it was kind of in a bad situation because we had this trial moving forward in TrialNet, but no company that was basically behind it. So that was a precarious situation.
Yeah, very precarious, but you still persevered, which is, whoops, let me just admit a few more people, this is really exemplary. And then I think, from there, prevention came onto the scene. We actually interviewed them in 2020, and they were kind of like, a digital company. They really didn't even have a base. And from there, do you want to just sort of illustrate how that, in a nutshell, progressed?
Well, so they ended up picking the drug up off of Macrogenics. And then shortly afterwards, we got to the endpoint of the prevention trial that TrialNet was doing. And it turned out it was, successful. And shortly afterwards, after the initial announcement of the results, they ended up, I guess, filing for breakthrough designation by the FDA, and they got that. And in addition, they filed for prime designation by the EMA, and then, then Brexit happened. So then they filed for innovation passport, which is what the term is called in the UK now, and they got that. So, the problem then became the original drug product that was made by Macrogenics was getting old. Yeah. And in fact, its expiration date was, last June. And so Prevention Bio needed to make a new lot of drug. And then with some initial PK studies in healthy volunteers, the FDA wasn't satisfied with the PK of the new drug compared to the old one. And that's where the complete response letter came in last July of 2021. And so after that, there was a fair amount of additional work that went on in terms of PK studies, PD studies, and so on. Eventually, that led to a kind of reconfiguration of what the dosing ought to be like. And so that's that's where things finally got to.
And so, really, the paper came out, the scientific translational medicine paper, March 2021, that was Emily Sims as the first author. You were on it, and others, Alice Long and Carla Greenbaum, et cetera. And that really, that also dug into some of the mechanism, right? That some of the partially exhausted effector cells were associated with the clinical response. And then maybe that also helped kind of push things over the finish line. Would you agree?
No, actually, I think in terms of the FDA evaluation, Monica, I think the mechanistic framework was important. I mean, clearly for discussion, and I think everyone felt better with some understanding of the mechanism. But I think really what kind of provided the substantial data was that there was data from the phase 3 trial that Macrogenics had done from the original trial that I had done, and then the other trials that I had done, and they all were showing the same thing.
Yeah.
All of the trials were showing that actually the drug would lead to retention of C-peptide, and that was even true in the prevention trial. I mean, that was in the Sims paper. So it was a very consistent story across, more than a decade of studies and many different clinical settings.
Yeah, well, I mean, ultimately, that's what really, showed that it was safe enough and worth any of the side effects. I mean, truly, if you speak to anyone who has type 1 diabetes or parent of a child 8+, right, getting, a little extra time or more, depending on, I guess, their recovery or their remission, whatever you want to call it, is really valuable and worth it. And, I think I wanted to ask your opinion, like, do you feel like this also opens the door to more exploration in further extending, adding other drugs, perhaps something
absolutely.
like that?
Yeah, I don't think, actually, I have a slide. Can I jump to my slide?
Yes, go to your slides. How many slides do you have?
Oh, I have, this is the important one.
Okay, good.
So, I mean, to your point and question, Monica, I think that's exactly right. I mean, this is just sort of the beginning of where we want to go. And the way I like to look at it is that if you so the data on the right is the data, at least as of last month, from individuals who are in the prevention trial. And you can see that there are some people who go way out. There's the first patient in the trial just was diagnosed with diabetes earlier this year. He had been out more than a decade from when he was first enrolled, and he was in high school when he was enrolled, and now he's grown up and he's living in New York and everything on his own. So the time here has some real meaning in people's lives. Absolutely. But some of the people, like again, as this graph shows, are doing extraordinarily well. And I think the important thing is to understand why those individuals are doing so well. And I should just mention my own bias is that the story that we have about inducing exhaustion of T cells, CD8 positive T cells, I think is interesting in terms of the acute effects of the drug. I'm not sure that explains the long-term efficacy, because those changes in T cells are gone after about a year of a single dose of drug. So I'm not sure what's happening there. But the way I like to think about this is that with those individuals, we're kind of where we were 15 years ago in checkpoint therapy for treatments of cancer. And if you take a look on the left is the one of the earlier anti-CTLA-4 papers, in which not everybody responded, but there are some people had very, very good responses. And that was a change. That was different than had been before, where melanoma, these are patients with melanoma. Melanoma was a fairly rapidly lethal disease, but now all of a sudden there was some signal of a response. And then gradually over time, there's more that's added to this. So now we don't just give ipilimumab, we give nivolumab, anti-PD-1, anti-PD-L1 agents, and then the curve changes. And there is a greater proportion of individuals who are responding, and they're responding for much longer periods of time. So that's exactly where I think we are now, just sort of at the beginning of where we need to go.
Yeah, and I mean, we've had discussions with Sarah Richardson's groups and others regarding the existence of endotypes. How would you? Do you feel that this is going to be an endotypal type of treatment, personalized medicine? I mean, some people, like even Matthias, will say, don't bring that up because it's not a good business case. But, it is something to consider. I mean, look at the example of breast cancer, right? At first, it was one size fit all. Now it's very finely, tuned. for the treatment, and the market supports that. So can't we say that this might happen here as well?
Well, it might be true. I'm not sure which we could speculate a lot on the endotypes, and potentially, there are some data that suggest that there might be, for example, certain HLA types seem to be particularly benefit the most from the drug. Certain autoantibodies seem to identify people who are most likely to respond. I think it's a little early to draw a conclusion about that, because those ends start to become pretty small, and they're not always reproducible across the trials. But there is one sort of endotype, maybe isn't the right word, but one aspect of this that I do think is important, which is timing. So, we've done trials with Teplizumab in people at risk with new onset diabetes. That's the ABATE trial here in the middle, and people who've had diabetes even for longer, who still have residual beta cell function. They've been diagnosed with diabetes after 4 to 12 months earlier. And you can sort of get the impression here, although it's not fair to compare the endpoints, that if anything, the prevention is working a bit better, and new onset seems to work pretty well as well. But if you wait longer, it doesn't seem to work quite as well. So I actually do think there is a window of treatment opportunity when this type of a drug, at least, this type of an approach is going to work. It could be that we could better define an endotype, but I think for sure we want to have some measure of autoimmune activity to know when to intervene.
Yeah. And so that'll be sort of next steps, identifying, that window. I wanted to, we're kind of halfway through. I want to encourage people to add questions into the chat, make this more like a conversation, or raise your hand to ask a question directly. So I'm sort of scrolling through people here to make sure I'm not missing anyone, but certainly all questions, all and any, are welcome. And I did want to ask you, so when we're talking about, this whole idea of the use of TZIELD, I guess it's being called, right? Is there an opportunity to use this in concert with eyelid implants, sort of like the one that the type that Vertex is developing?
Yeah, it's amazing, Monica, because you know all my slides and we didn't talk beforehand.
No.
So I think that's exactly right. That's exactly what we ought to be doing. Creating beta cell replacements. Perhaps conditioning, if that term's okay, conditioning a recipient with anti-CD3, and then putting in those beta cell replacements. I guess my thought on this is that the drug would stop the recurrent autoimmunity if it was to occur with the beta cell replacement. But I would also point out that as an anti-CD3 antibody, it works very well. Even in sort of conventional settings, such as the original use of this drug was in patients who were receiving kidney or kidney pancreas transplants. There's studies from Bernard Herring about its use in patients who've gotten eyelid transplants. So I think it works there. And I also think it works in terms of stemming autoimmunity.
All right, that's good news. I have a question from Peter Butler at UCLA. Would you like to define, well, he said, congratulations. Kevan, would you like to define for clinical practice as to how we identify the patients for this RX?
Yeah, so that's the challenge, Peter. The way TrialNet did it is relatives were screened, and then we found people who are double autoantibody positive and dysglycemic, and those were people eligible for the trial. I mean, you needed to have stage 2 diabetes, which includes both the immunologic markers and some sort of metabolic impairment. However, I do think it's time, and I'm really glad I put these slides in here. Sorry, I do think it's time to start thinking about, should we screen the general population? Because, this work from Annette Ziegler on the left, where she screened individuals in Bavaria, identified a number of children who are even double autoantibody positive, and their rates of progression to diabetes are similar to relatives who are shown on the right over the boxes a similar period of time. So, I think now that we have something to do about it, looking for individuals who may potentially benefit is a reasonable course.
So you're thinking this is sort of just part of well baby checkups?
Yeah, I mean, it could be well baby checkup.
Yeah, why not? Right.
Right.
I mean, I think
we screen for a lot of stuff.
Yeah.
That's less common than diabetes.
And less dangerous.
Yeah.
I have a Decio Zurich from ULB. He has a question. Decio, do you want to unmute?
Hey, Kevin. Congratulations.
Thank you.
This was finally approved. I have a question about patient selection, because there was, Monica was making this comparison with breast cancer, but in the case of targeted therapy in cancer, you have the opportunity to obtain tissue, look at gene expression, and then define which type of therapy I'm going to use. In the case of type 1, we are limited to what is circulating in the blood. And I think this has been a little bit disappointing up to now. So you still have hope that by looking at circulating T cells or other markers, we can obtain indications of which patient would benefit most?
Unfortunately, I'm not sure that immunologic markers are going to be useful here. And, we always get into this argument about it. And the argument usually ends up silencing the discussion by the point that, well, probably the T cells that you're interested in are in the islet. So, that's the end of that discussion. On the other hand, and I have to say, my lab was very interested in developing ways of identifying beta cell killing in vivo, and that has not turned out to be a very useful approach to identifying who might be eligible. I mean, there are a number of reasons why I think that type of an assay isn't particularly useful for screening individuals. I would think, frankly, that short of some sort of a marker of beta cell killing, the best thing that we have right now is metabolic function, unfortunately.
Okay. Here's another question. Were there any microbiome markers / changes as a result of treatment? Microbiome is a big place of inquiry in the context of autoimmunity, and even now in type 1 diabetes, people like Amra Altinus over there in Boston College are digging into that.
Yeah, that's a great question. And I really, really wish we knew the answer to that, because I'm sure there's gold in there, but we don't know. And the only thing I can tell you is that we did some studies of the same anti-CD3 antibody in humanized mice, because and where we looked at that question, we had data from my lab and Richard Flavel's lab that the way this induction of an exhausted phenotype seemed to involve migration to the gut wall. And so the question came up, well, does the microbiome have anything to do with that? And the data we had from the humanized mice that we treated with antibiotics was absolutely it did. Because if you treated the mice with antibiotics, the anti-CD3 didn't work. So I think there's something really important there. That would be my guess. We don't have any data about this, unfortunately.
That's very interesting. We've invited a couple representatives from Finch, which is a biotech company that focuses on the microbiome, and they have some interest in autoimmunity. So maybe they'll pursue that.
Yeah, there might be endotypes that can be defined in that way.
Yeah, that's a really interesting, idea. Sorry, I'm scrolling through here. Any other questions? Are there any other questions here? There's a lot of people here. I would say, when we talk about one sort of practical piece, and how are patients going to be able to afford this? I mean, we can see in the press releases that it's going to be quite expensive for the 2-week treatment, right? I know there's a COMPASS program in place to help patients get access with the patient assistance program, but what are your thoughts around that access to this?
Yeah, Monica, I've had absolutely nothing to do with that. And, I think that's probably good for the company that they don't ask my opinion. I'd be giving it away.
Yeah.
So I'm not in that business. I don't know what, I know what has been said about what it's going to cost. I don't know what, obviously people aren't going to pay that. We can get into a whole political discussion about what it should cost and so on, but I don't know. And I also don't know how you would compare it to costs of other biologics. So
right. I think it's right there in the same ballpark with other biologics. Yeah. And, as I've said, they have a program in place to help people gain access. So I think that's very promising.
I have to say, I should just mention the part of the reason I found that news, and it was news to me last week, upsetting, is because there are many, we've and I said this many times in my role in TrialNet, we have a very distorted bias in how we do clinical trials, and there are many underrepresented groups that don't participate in our trials and don't get access to this, the developments from our research. So I think that's an area that needs work. Yeah,
agreed. I would also wonder, if in terms of its, extension into other groups. I mean, are people starting, other clinical trials sort of to piggyback on this? Are there conversations, or is there anything tangible that's happening? What's the word on the street?
Well, I can tell you in TrialNet, what we're interested in doing are a couple things. One is we'd like to do the company has to do a trial in individuals under the age of 8. And in TrialNet, we're also very interested in that. So we're keen to start that trial. The immune repertoire of very young children is different than people who are in their mid-teens or 20s. Yeah. So I think, how and whether the drug will work in younger individuals is an important biologic question to answer. The other thing is, I emphasize this need to, this thing about the window, but there are people who have stage 1 diabetes who don't have any metabolic impairments who appear to be progressing rapidly. And if we can identify those individuals, they may also benefit from this type of treatment.
Need a lot more sort of boots on the ground in the laboratories and money to fund this, I would say.
Yeah, one last thing, Monica, I should have brought it up. The other thing is, and particularly since Sergio's on the call, one of the trials that I'm very excited about is to combine it with a TYK2 inhibitor. Now, obviously, the TYK2 inhibitor has effects beyond just beta cells, but I do think, particularly in people who already have diabetes, combining those agents, to me, makes sense.
What do you think, Decio?
I think it's a very good idea. The other thing that I think would be interesting to consider is lifestyle intervention. I think there are data to suggest that a planned lifestyle intervention with physical activity can protect pancreatic beta cells, especially against endoplasmic reticulum stress.
Well said. Well, it looks like Matthias Von Herrath has joined us just for the very tail end. Matthias, would you like to contribute or have anything to say? I know you're on vacation, so I appreciate you dialing in.
Oh, I'm so sorry. I had it for 10:30 this morning.
No worries. Well, we had a good discussion. We spoke about, the advent of the first drug approved by the FDA for stopping progression to stage 3 of type 1 diabetes. And we talked about the history of Teplizumab and, sort of what's ahead. Is there anything else you'd like to add?
Only combination therapy with beta cell centric components, that would be nice. And it's a great achievement that we have this now. It should be great for the whole field. That's short and sweet. How about that? I'm sorry, I thought it was an hour later. But
No
maybe
worries.
because of my travel and the time change, that's what happened.
No worries at all. I think someone else is saying, microbiome response is fascinating. Could you perhaps integrate a microbiome screening into preventative early childhood screenings to track changes over time? Yeah, some, screening kids from the beginning and looking for the way they progress and potentially endotypes would be, I think, would inform the knowledge base for sure.
Yeah.
Okay, in the interest of time, last call for questions. Anyone? All right, I guess you can reach out directly, and this will be recorded and hosted on our YouTube site, so for further watching at your leisure. And thank you again for joining us, Kevan. It's just a phenomenal accomplishment to get this to market. And I also think, as your comments showed, it really just it does take a village, right? It really does. All different types of scientists working together and interfacing with the FDA. And that's no easy challenge. It's incredibly challenging to get a drug like this to market, and just hats off for all that contributed to that. So thank you again. And I hope you have a great rest of your day and a good Thanksgiving.
Thank you very much.
Thanks
Bye.
again.
Sorry,
Bye-bye.
Kevin, I was late again.
No problem. We'll talk.
Talk again. Bye-bye.